Thank you, Lynn. Next up is Meg Ehm, who's the Director of External Strategic Alliances for Genetics at GSK.
Good afternoon. All right, today I'm going to speak on behalf of an emerging consortium that is focused on using human genetic evidence to identify and develop medicines that make a difference in people's lives. This work is built on a lot of what we've been talking about today, the technological advances, the understanding of the biological pathways, genetics, genomics, the progress in gene editing, and most importantly, and why we're all here today, the emergence of the large-scale human health databases. Last fall, our group came together to discuss the industry view of what an ideal genomic bioresource offering and the models that would allow us to use the bioresources to support target identification with an eye towards clinical development, patient stratification, and pharmacogenetics. Our observations were that previous efforts that assess the impact of genetic variation on clinical phenotypes have used population studies and genetic analysis consortium. But we felt that often those efforts had reduced the phenotypes to a common denominator and rarely had longitudinal information, that very few collections had drug usage or drug response information, that the research was slow, and that many conditions hadn't been studied. I provide an example that illustrates the kind... Okay, sorry. I provide an example that illustrates the kind of research that we would like to be able to do, but also the challenges that many of us have faced. And actually, there have been several great examples already given earlier today. At GSK, we identified a rare missense mutation, the alanine 316 threonine, and the GLP1R gene in a sequencing study. And when we combined the results from that study, genotyping additional samples in collaboration with academic collaborators and in silico results, this variant was found to be associated with fasting glucose. This gene encodes the glucagon-like peptide 1 receptor, the target of GLP1R agonists. And while we observed that similar to GLP-1R agonist therapy, the minor allele was associated with fasting glucose, it was not associated with type 2 glucose or insulin. Regulatory authorities have indicated that new drugs for treating type 2 diabetes should not be associated with an unacceptable risk in cardiovascular risk. And we were particularly, since this variant seemed to have some mimics to the drugs that were targeting this receptor, we were particularly interested in whether or not the variant was associated with disease outcomes, including type 2 diabetes, coronary heart disease, and pancreatic cancer. We observed an association with a lower type 2 diabetes risk consistent with therapies that targeted this receptor. And the minor allele was also associated with a lower risk of heart disease, thus providing supportive evidence that GLP-1R agonists are likely not to be associated with an unacceptable increase in cardiovascular risk. Unfortunately, we did not find evidence of a significant association in either direction with pancreatic cancer, although you can see wide confidence intervals. Our results provided an encouraging signal that these agents may be associated with benefit, a question that was currently the subject of clinical trials. This work was supported by a press release made last week by Novo Nordisk, who reported that their GLP-1 agonist had been shown to reduce the risk of major cardiovascular events in the LEADER trial. So we're very excited about the potential to use variants like this one and others that were discussed earlier today to understand drug targets and their on and off target effects. However, the completion of this work that I described was daunting. The analysis of coronary heart disease involved more than 30 cohorts, and the study of fasting glucose involved more than 50 cohorts. And you can see that the samples needed was 240,000 for the study of the cardiovascular risk. So we're very excited about the potential to be able to study genetic variants with on and off target associations, but the scale of international collaboration to do this work is daunting. And so the question is how can we speed this up? How can we be able to do more studies like this that we think will really make a difference? We were very excited about the potential to use the EMR-linked biobanks to influence, to do this type of research. And I think in addition to many of the types of advantage that we've talked about here already, I think some particular ones have emerged. The ability to be able to study conditions that are not generally available, like RSV hospitalization, or being able to really understand disease trajectories like heart failure that turns into pulmonary edema and perhaps renal conditions. We're interested, as we came together in this consortium, we talked about how we envision using EMR-linked biobanks, the ideas of recruiting patients by their genotype or phenotype, the ability to be able to study the effects of gene variants on clinical phenotypes, which can anticipate the effects of drug treatment, or even the ability to study the effect of gene variants on the trajectory of disease, and then perhaps use the use of the healthcare system to support indication optimization. So as we discussed this work through last fall, we came together and really said we see value in these integrated medical and genomic resources to identify and prioritize drug targets and understand drug response. And I think, like many, we identified these 5 key factors: comprehensive and diverse health data, the genetic data, biological sample availability, informatics, and the ability to recontact patients. Through our discussions, the consortium came together around a couple of key tenets. And, you know, on the one hand, very excited about the potential for EMR-linked biobanks, but also frustrated with the models and the progress thus far. So together we came to, almost in the first meeting, I think, we said, we view the developing research-enabled environment as a pre-competitive activity and will require more resources than any one company can provide. We agreed that it was critical to engage early in the design and the development of genomic EMR resources to leverage the unique insight that we have in the use of these resources for drug discovery and development questions. And then we, I think, importantly agreed that the harmonization of multiple resources across ethnicities, across health settings will be needed to fully realize the value. No one resource will be enough. We really wanted, as we talked through how we wanted to use these resources, the key principle that came up is there was data and capabilities that are needed related to patient data that would be important to address the gap between disease-associated loci and a therapeutic strategy on a target-by-target basis. And we envisioned that an industry-led effort that would collaborate and be in partnership with EMR-linked bioresources could enable these scoping and execution of studies without compromising the academic goals as well. And we came together and said, this is something we want to make happen. So we first discussed exactly the kinds of questions that we wanted to be able to ask. Our discussions have highlighted our excitement about evaluating genetic variants with outcomes, kind of like what I talked about in the GLP-1R examples. But quickly we wanted to move toward the ability to look at a genetic variant associated with disease progression, complications, and ultimately we see a resource that predicts the outcome progression that modulating a drug target to be game-changing. And we're anticipating the use of systems biology or pharmacology with genetic and frequently sampled genetic and biomarker data would facilitate this shift. Almost at the same time, we discussed what would be important on the clinical trial side, not so much to facilitate development, but very importantly to allow us to do the early clinical studies. So we considered the use of EMR-linked biobanks to be able to recruit patients by genotype and phenotype so that we could really understand the differential rates of progressions and exposures. We sat down and we started to put together emerging requirements, things that we felt like would be really useful for asking the particular questions. And we agreed on size and scope, the amount of health care data, the informatics capability, and the biospecimen and recall. But certainly see this as emerging requirements that will include continued discussion. So what we're hoping to do is to to establish a consortium that would focus on being able to assess if a target is associated with clinical conditions and drug response to enable the prediction of possible outcomes. We're looking at what would a research-enabled environment look like, and how could that facilitate drug discovery and development. We understand that a consent and infrastructure will be needed to make sure that we could identify and recontact patients for clinical trials of all types using genetic and phenotypic information. It'll be critical that there's an access model that not only protects the patients or the participants, but also enables the scoping and analysis of the data. Certainly informatics capability that enable the harmonization of phenotypes and meta-analysis across studies will be important. And certainly we could imagine that it might be useful to be able to leverage existing or generate additional data for all or subsets of the resource. The next steps are to assemble parties that are interested in participating in the consortium organization and planning phase. We want to work together to answer the question, what are the models that would bring together EMR-linked biobank resources to enable us all to achieve our goals? And we intend to develop a business plan and a funding model to realize these goals by the end of 2016. We feel that our driving principles, no one resource is enough. We cannot do it alone. Much of this work is pre-competitive, as well as the interest from emerging biobanks and the lack of focus from the public efforts on the use for drug discovery and development point to a value in working together to answer the question, what are the models that would bring EMR-linked biobank resources together to answer questions of interest? Thank you.